damage of cardiac cells. And the three that we chose to look at are iGEM, CRP, and myosin heavy chain. And the receptors for those that we expressed on our stem cells are LFA1, Fc gamma, and an anti-MHC antibody. Because we started really late in the summer, we only started our cloning at the end of August, and we had to design all of the parts ourselves. We decided to do PCR fusion to accelerate the production because subcloning would have taken us too long. So I will talk about the iGEM LFA1 signaling, and they'll talk about the other ones. And so the reason we used iGEM is because when it is bound to the membrane, it forms a dimer with two active binding domains, and with that, two LFA1 molecules can actually bind to the same molecule and be very, very close to each other. And this proximity allows us to use the synthetic split signaling system that we decided to use, which is split ubiquitin, which I'll go into a little more detail. But because it's so close, it'll be able to interact and release the transcription factor that turns on our effectors. So the reason we know that LFA1 is close enough is through a complex experiment that was done by the Springer lab at Harvard. And basically what they showed using threat analysis is that that LFA1 molecules are close enough to each other to have a fluorescence resonance energy transfer. And because they're close enough for that to occur, that means they're close enough for their intracellular parts to interact, allowing us to use the split ubiquitin system. And so the basic idea behind the split ubiquitin system is ubiquitin, whenever it's found normally in the cell, will get cleaved by ubiquitin protease. And people have used split ubiquitin. Basically, they separate the ubiquitin into a C terminal called cub and an N terminal part called nub, and will fuse it to two proteins that they believe interact. And if the two proteins interact, the cub and the nub are close enough together, they will reconstitute the ubiquitin construct, and which will get cleaved and release the transcription factor. And the transcription factor that we used is LexA VP16, and LexA is a DNA binding protein that's derived from E. coli, and VP16 is a transcriptional activator from herpes simplex virus.
So the second system we designed is derived from the endogenous. Our second signaling system is derived from the endogenous system found in the cyanobacterium simplex. So basically, we looked at the DNA E gene, which is split up into two pieces that are almost 745,000 base pairs apart. What's more interesting about these genes is that is the way that they get reconstituted after translation. So the DNA end portion is made up of an extine N region and an intine N region. And the DNA EC region is made up of intine C and an extine C. After translation, the two intine regions come together, splice themselves out, and splice the extine regions together to form a full DNA polymerase supplement. And our system takes advantage of this post-transcriptional splicing ability to produce a functional transcription factor only after intine interaction. And I'll talk about that more in a little bit. So our synthetic system is focused on the signaling effect of C-reactive protein via an Fc gamma receptor. Human CRP is a plasma protein that is generally found as a pentamer in human plasma and has been shown to be significantly upregulated after myocardial infarction, especially within the first 50 hours after the infarct. It has also been found to be homogenously deposited on inflamed tissue. Second panel. Both of these attributes make it a really important inflammatory marker, as well as a good therapeutic target for our immune system. Although the full signaling system is quite complicated, we took advantage of the initial interactions to drive the production of our signal. Specifically, we focus on the interactions between SYK and PD5, and I'll go into that in a second. Essentially, replace the endogenous endogenous XTEN region with LEX-A and the XTEN-C region with VP16. We then link the LEX-A intine construct with PD5 and the VP16 intine C with SYK. That's the description of the whole system. Thus, after CRP binds the Fc gamma receptors, cross-linking receptors, we get phosphorylation of the intracellular activation motifs. This in turn recruits various proteins such as SYK. After SYK gets phosphorylated, we get recruitment of other proteins such as PD5. This close interaction between SYK and PD5 allows the two intine sections to interact, producing full transcription factor that we use for later activation of our downstream circuits. Although the system has some disadvantages, since there is a basal presentation of CRP in the plasma, there are also, which could interfere with signaling, and the system in stem cells and myocardicytes as an unknown effect. There are some major advantages to the system. The widespread usability and adaptability is the main one, since CRP is generally upregulated in almost all inflammatory tissue. And as you just said before, these signaling systems are endogenous and proven to work, and the use of the LEX-A VP16 system allows for limited potential for crosstalk.
Okay, I'm going to talk about our third and final signaling system. So when we wanted to find a target that had high specificity, and one of the things that we found was when a patient undergoes a myocardial infarction, that the intracellular MHC is actually exposed in the cell surface, thereby making it an excellent target. Our third construct consists of a human anti-MHC single-chain variable fragment, which is fused to the extracellular erythropoietin and transmembrane portion of the erythropoietin receptor, and then fused to the intracellular domain of GP130. This MHC system also uses the same signaling system as the CRP system, which is the split intine. Well, the receptors have the DNA EC, which is then fused to the VP16. Endogenous GP130 is usually closely associated with Janus kinases, and upon binding to MHC, the receptors homodimerize, and the JAKs, the Janus kinases, phosphorylate certain tyrosine residues on the cytoplasmic portion of GP130. These in turn provide the docking sites for SH2 domain-containing proteins. And one of these proteins is SHP2. And being so, we chose this as our second half of our split intine system and fused that to the second half of the split intine, DNA EN, and then LEX-A. This is the same thing as the CRP system and will reconstitute in vivo the transcription factor of VP16 LEX-A. Our anti-MHC scFv has high specificity for human cardiac myosin heavy chain and has little affinity for other things such as actin and BSA. There are some obvious advantages and drawbacks to the system. So first, as I noted, this has high specificity because it's coming from a human monoclonal antibody, the variable heavy and variable light region. This has high specificity for MHC. Also, this system almost has no background. Because the MHC is usually not exposed on the outside, and therefore the receptors can't bind to it on normal cells, this has obvious advantages over the other two systems. But the most obvious advantage is that because we're using an scFv, you can easily adapt this system to target, say, cancer, Alzheimer's, other things by simply changing the scFv and changing the effectors. And some of the drawbacks is, as they stated, because we're using parts of the endogenous pathway, there might be, SHP2 elicits an inflammation and growth signals, and that might cause some background. So in all three of the systems, the switch is the LEX-A VP16. Some of the effectors, some of the considerations we took into account in designing this experiment was that, as Anastasia had said before, when current stem cell, current myocardicytes therapies, the major problem they have is when the cells are targeted to the damaged area, the cells the cells either undergo necrosis or apoptosis. So we used inhibitors of both. We used the BCL-XL, which is part of the BCL2 family of apoptosis inhibitors. We also use P35 and IAP, which inhibit the caspase pathway of apoptosis. And then we also use 2 necrosis inhibitors, Serpin-6 and calcistatin. And these are all under the control of the LEX-A, so these won't be on unless activation of the receptors. Another problem that we had to address was how do we get the cells to integrate actually into the damaged heart tissue. So we overexpressed a cell-cell protein, which is NCadherin, and in studies it shows that if you have, say, a disproportionate amount of NCadherin expressed on 2 different populations of cells, one having more, one having less. The green expresses more and the red expresses less. You can see that over a 24-hour period, the cells actually localize to the middle. We hope to use this kind of information so that we may target our cells to actually the inside of the damaged tissue. Another problem that we had to address was, as Anastasia said in the beginning, when the heart undergoes an MI, the blood vessels become damaged and they no longer support nutrients or oxygens to that tissue. So even if we would replace cells, they still wouldn't be able to fully function. So we added a VEGF, which induces angiogenesis. Another thing we use is HGF, hepatocyte growth factor, which inhibits fibrotic scar formation. So we have some preliminary studies. We used the CRP signaling circuit. We treated H9C2s, which is rat cardiomyocytes with hydrogen peroxide, and this induces an MI-like effect. We also transfected 293T cells with either all or part of our CRP circuit, and then we added those transfected 293Ts to the damaged cardiomyocytes. So in the control, we just put Fc gamma, which is only one of the 3 receptors for the CRP system, so there should be no effectors activated. In this panel, this is red cells. The red fluorescence shows that the cells are transfected. There's almost no background in the green. We used our LEX-A operator with GFP, so there should be no signal, and the only signal that you do see is leaching from the highly fluorescent red cells. In the full CRP circuit, you see that there's red fluorescence, meaning the cells are transfected, but there's also green signal, meaning that there must have been activation of the signaling circuit, and therefore the downstream GFP effector is also activated. We also have some very, very preliminary studies on expression of NCAD and on 293Ts and the penetration of them into P19 differentiated embryonic bodies. So we hope to test our cells further. We didn't quite have enough time. We wanted to test the specificity of each of our cell signaling systems. We'd also want to test our effectors, the apoptosis, anti-apoptosis pathway, the NCadherin cell sorting effectors, and then also the restoration of the microenvironment, the VEGF and HGF. Of course, we want to test this in vivo. We've only done in vitro studies. In vivo, we'd use a mice or rat MI, where we would ligate the coronary artery to produce an MI-like effect and then introduce our cells. So we don't want this just to be an interesting iGEM summer project. We'd like this to have real-world therapeutic applications. And so there are obvious problems with any kind of system like this and a lot of the iGEM systems which require gene therapy. So gene therapy has inherent complications. And so one of the ways we would circumvent this with our system is actually induce a transient expression of our systems using the herpes simplex virus 1 amplicons. And even though it's a transient expression, it's perfect for our system because we only want the cells to express it transiently and then integrate. And then whether or not they express the genes afterwards doesn't really matter because they will already have integrated into the damaged tissue. Also, we don't really know which of these cells work best in vivo, and so in that case, we want the rat and mice testing would show us maybe we need multiple ones. Maybe we need to create an AND gate to increase the specificity and targeting of our cells to the damaged location. And then lastly, we'd like to use of course, our choice of target cells would be autologous adult stem cells, but for that to work, we also need our differentiate circuit, which will differentiate them into cardiac cells. So we'd also like to thank our sponsors, Abbott and Panorama Research, and we hope that people can benefit from our therapy in the future. Questions?
So you mentioned the sequential circuit return on sequentially. In the design for that, don't you guys build it in a way where you'd be able to sort of drop in different genes? Is it kind of hard to do for?
Oh, is it like a BioBrick system?
Is it BioBricks or is it just in general amenable to putting in other genes?
Everything is flanked with precision enzyme sites, so it's easy to pop in, pop out. But though they may not be compatible with the BioBrick system because of them, there was issues because we had so many different genes and we couldn't use the same restriction sites, so they might not be compatible.
So is the idea that you're engineering the stem cell, you get the stem cells out of the person and then add genes and then put them back in the person and then they go into the heart and fix it?
That could be one possibility.
We'll come back to that.
That would probably be the most reasonable one. I mean, you can get adult stem cells, from the iliac crest, from the bone marrow. And, people do that nowadays with, the current therapies that Anastasia was talking about, how you can use bispecific antibodies, and it's been done on patients already. So it wouldn't be a problem. The biotech things are already in place. So
And so then the idea is that you would just engineer the cells.
yes.
And then put them back in. The nature of the engineering, as I understand it, is that it would increase invasion of the cells, increase angiogenesis, and increase survival,
Yes.
right? And so it seems like a formula for making metastatic cancer. Is there any worry about that?
Well, that's why we wanted to have a transient expression. So once they turn on, for a short amount of time, they would turn off. I mean, it's not, if you had constitutive expression, then that would be a big problem. But because we're using a transient expression, it's less so.
We'll come back in. Okay.
Kind of a naive question. Can the split ubiquitin system work in prokaryotes, or is it just specifically for eukaryotes?
Actually, it was first shown in prokaryotes, and then in yeast, and then there's a recent paper that was shown in mammalian system.
So it's only eukaryotes.
Any last questions?
Sort of a tangential question. What does peroxide do to cause an MI phenotype in your cultures?
It stresses the cells in the same way. We didn't add the slides, but you can see that the cells undergo kind of like a hypoxic effect, and you can see that the cardiac cells will have stress fibers. A lot of their nuclei will look damaged and things like that. We didn't show all of our data, but there's actually, if you use different concentrations of hydrogen peroxide, they will induce either total heart damage or just a stress-like effect for MI, and we tested both of those, but I don't know.
What's the range of peroxide?
So the range is 50 micromolar to about 200, and for the damage, it's 150. They're both different times, 150 micromolar, and the other one is 100 micromolar.
You showed one cell that was actually producing the GFP. Was the transfection efficiency low?
Yes, and that was because we had the DNA quality was at different levels, and so yes, they were at different levels, and so that'd be one thing that we address when we go back. We try to make cleaner preps, and so we'd have higher transfection. We also transfected cardiomyocytes, the H92Cs, directly with just the genes, but we tried different transfection reagents, and they didn't work. They didn't transfect well.